I'd just like to thank you all for joining us to the next of the MSc webinar series. So this one's called, it's based on ELISAs and multiplex ELISAs. So for any of you that weren't at one of our previous presentations, okay. Just give you a brief introduction to everyone that's on the team. So my name's Lisa. I did my PhD at the University of Leeds. I've got 7 years postdoc experience. Please do not try and work out how old I am. And my main techniques are molecular biology, protein expression, Western blotting. We have Paul, who did his PhD in Galway. He's got 3 years postdoc experience. His main techniques are flow cytometry, cell culture, and ELISA. And last but not least is Emma. She did her PhD in DCU. She's got over 2.5 years postdoc experience, and her main techniques are cytochemistry, histochemistry, flow cytometry, and Western blotting. So that's the three of us. Hopefully some of you know us, which would be good. So I'm just going through what I'm going to be talking about today. So I'm going to give an overview of ELISA technology, the different types of ELISAs, how to analyze them, sample preparation, the importance of your CV data, give you some tips on doing your ELISAs, a new technology that we have from a company called ultralow detection, which is quite cool. And then I'll go into multiplex, which is kind of split into two different types, which is one is flow cytometry based and one is membrane and slide based version. And then I'll just go into a recap, and in case any of you have loads and loads of questions. So ELISAs, what does it actually stand for? ELISA basically stands for enzyme-linked immunosorbent assays, and they're used to basically quantify a desired marker. So that marker could be either hormones, it could be proteins, it can be all sorts of different things. And what it can be used for is it can serve as either an orthogonal or an alternative assay to Western blotting. Where it betters blotting is the fact that it's usually, it is faster, it's highly sensitive, it's also high throughput. Like try to do, 2 ELISAs compared to probably about 60 Western blots. It's also very reproducible and it's very flexible. So just give you an example here on the right. What we have here is a Western blot of EPO compared against beta-actin at increasing concentrations of EPO. So you see the intensity get higher. Obviously, you have to take into account the beta-actin is a bit lower here. And then compare that against the ELISA. So you've got the ELISA down here. And as you can see, as you've got the increase of EPO, I hope there's no Lance Armstrong fans in the audience. Might be his samples. So just to go through what ELISAs actually do and how you actually use them. So all ELISAs are basically boiled down to basically 3 things. You have your antigen, so this is your targets. This can, like I said, could be hormone or protein. And then you have your antibody. And the antibody can be conjugated to HRP or a fluorophore or a luminescent molecule. Basically, you can add a substrate and then you'll get your signal. So from that, typically ELISAs are done in a 96-fold plate. So this means the more antigen you have, the higher the intensity will be, because obviously you've got more antibody binding to it, so you've got more signal. Obviously, if you've got less, then your intensity would be lower. Typically, you'd run standards. In this case, I've demonstrated here running the standards in duplicate, which I think is the standard way of doing ELISAs. Never run it in singlet, and triplicate is obviously regarded as the better method, but obviously you'll be using much more of your plate. And from your standards, you can then calculate your standard curve, which will then enable you to calculate the concentration of your protein, which I'll go into more detail in a bit. So, like I said before, the essential components of ELISA is your antigen you're trying to detect, and a specific antibody, and then a system to quantify the concentration or measure how much antigen you have. So there are 4 main types of ELISAs. There's direct, indirect, sandwich, and competitive. And I'll go through in more detail on each of these. and where their pros and their cons are. So the direct ELISA was developed by Perlman and Engvall. This is usually used to detect high molecular weight antigens, and in this case, your primary antibody is the one that's labelled with either the fluorophore or the HRP. So what you'd have is you would have your antigen coated onto the bottom of the well. You then add your primary antibody, which is then conjugated. You then wash and allow the substrate to incubate, and then you read. So some pros for this: obviously it's quick because you're only doing one incubation with just the primary antibody. There's no secondary cross-reactivity with the secondary antibody. It's also a really good option if there's no commercial ELISAs available. Some cons for this though, obviously labeling of the antibody can be very time consuming, it can be expensive, and you typically get minimal signal amplification, and labeling may affect the immunoreactivity to the antigen as well. So indirect, this is basically just a build-up of the direct method. This uses a two-step binding process. So this is where your primary antibody is actually unlabeled, and you are detecting the signal using a labeled secondary antibody. So it's the same as the direct version where you have your antigen coated to the bottom of the well. You add your primary antibody, which will obviously recognize the antigen and bind to it. You add your secondary antibody, and then wash and add substrate and incubate, and then you read. So some pros for this: obviously there's a wide range of secondary antibodies available. You've got obviously murine, rabbit, whatever you basically raised your primary antibody in. It's very versatile. You can use many primary antibodies in one species and use the same secondary antibody. You've also got maximum immunoreactivity, and in terms of sensitivity, is obviously increased as well because each primary antibody would contain several epitopes that can be bound by the secondary antibody. Some cons to this though is some cross-reactivity might occur, and you typically find it's longer because obviously you've got more incubation steps. This assay is typically used for people that are using something to identify bacteria, parasites, or viruses. So I'll move on to probably what is most commonly known, the most commonly known ELISA. So this is what most people think of when you say an ELISA, and that's the sandwich ELISA. This uses match antibody pairs, sorry. And this is where each antibody is actually specific for a different epitope. So what do I mean by that? So here's your protein. You'll have your primary antibody or a capture antibody that will bind to one part of the protein, and then you have your detection or your secondary antibody, which will bind to a completely different part of the protein. Your capture antibody is coated onto the wells, and it's your secondary antibody or detection antibody that is conjugated. So, like I said before, you've got your primary antibody coated onto your surface. You've got your mixed sample, so you add this to your well. Your antigen will bind to your capture antibody, and you can wash away anything that's non-specific. You would then add your detection antibody, so this is your secondary antibody, incubate with substrate, and then you can read this. So some pros for this: it's got high specificity, it's suitable for complex samples, it's very flexible and it's very sensitive. Obviously, the cons are it's longer because you've got more incubation steps and wash steps, and obviously you need antibody pairs. So competitive ELISAs, which are also known as inhibition ELISAs. So there's actually a couple of different versions of what a competitive ELISA is, but this is the version I've chosen. So this assay uses an inverse correlation between antigen concentration. So what I mean by that is, as I said before, the more antigen you have, the higher the signal. But in this case, you've got the reverse. So the more antigen you have, you'll have less signal. So I'll go through that in a little bit more detail. This assay is usually used for detecting low molecular weight antigens, for example, hormones, or for those samples that got limited number of epitopes. So what you have in this situation is you'd still have the primary antibody coated onto the surface. Now in the next step, you actually add a mix of your sample with your target antigen, but conjugated to a signal. And this is of a known concentration, so you know how much is in there. You then wash this, and then obviously the antigen and your antigen that's conjugated to a signal then binds onto the capture antibody. So what's happening here is, so if you had more antigen, if your sample had more of your target protein, this would outcompete the conjugated version. So this means your signal will be a lot less when you go to read it. So some pros for this, obviously, like I said, it can be used for low molecular weight antigens. It's quick, you've only got one incubation time. And obviously you're removing the secondary antibody, cross-reactivity, and it's got high sensitivity. Some cons for this, some immunoreactivity of the primary antibody may be affected because your antigen has been labeled. The labeling of the antigen will obviously take time, and obviously it's more difficult to calculate the results, but there are programs such as GraphPad Prism that can be used to help you calculate competitive ELISAs. So one of the important things in your ELISA is obviously making a standard. So you generally create what we call a serial dilution of your standards, and you usually do a 2-in-1 dilution. So this means you have your stock concentration, and what you do is you take a portion of this, say 200 microliters, and then you add 200 microliters of buffer or water, usually buffer, and then you basically make that serial dilution as you go down, and then this will produce your linear graph, basically. So here you've got your decreasing concentration of protein, and then obviously your decreasing level of OD. Okay. So using your standard curve, you can then use that curve to analyze your ELISAs. So ELISAs can actually be split into 3 types. So one could be qualitative. This is a determination of whether your antigen is either present or not. So for example, say you've got a knockout and your wild type, you want to see whether that protein is present or not. Semi-quantitative, so this is a relative comparison of the antigen levels between samples. So say you have stimulated your cells with a ligand, and you want to see if the protein levels have increased or decreased. So this could be semi-quantitative. So has it increased since you added the ligand, or has it decreased? And then there's quantitative. So this is actually a calculation of the amount of antigen in your sample by using the standard curve like I showed before of known concentration of antigen. So how do we do this? So as I said before, you've got your plot of your known protein concentration against the OD of the values that you saw in your assay. You can do this in Excel, or you can do this in Prism. I always did my ELISAs in Excel for some bizarre reason. So what you do is you create a scatter graph to begin with, and then you can add a trend line. So here's your trend line that you've added. Now your trend line will actually, in Excel, your trend line will actually give you a lot of information. So it will give you the equation of your trend line, which is very important to use in calculating your protein concentration, and then the R-squared value. Now the R-squared value is basically demonstrating like almost the linearity of your graph. So an R-squared value of 1 is obviously the optimum and very, very difficult to obtain, as you probably can figure. But something of a value of 0.998, say, and above is very good. Excuse me. So it's always important to know what your R-squared value is, because this can actually give you a lot of information and depend on how your assay has gone. Because if your R-squared value is lower, then it might be something where you address and you think, right, I need to run this assay again, because it means that the concentration that I'm going to calculate is not going to be correct. So you might have to change the standards that you're using in your graph to make it more linear, or just re-prepare them. So how do I calculate the concentration using the standard curve? So, say for example, we got an OD of 0.7 on my graph. So what I do is I draw a line from 0.7 across the trend line, and then from there move downwards, and then that will give me my protein concentration of 35 picograms per mil. Excuse me. So that's one way of doing it, but then you can also use, like I said, the calculation of your trend line. So using the equation, you basically put in your x value, which is your y value, which is 0.7, and from that you can work out the concentration is 34.997 picograms per mil. Obviously, this rounded up is 35 picograms per mil, but obviously this is much more accurate. So sample preparation. Now, there are actually many different samples you can use. I've given you a selection, I think, of between 6 and 8 samples. I'm not going into a huge amount of detail about how to prepare them, but if you have any questions on this, then please just contact myself, Emma, or Paul, and we'll be happy to pass on some information on that. So just to go through some of the main examples of samples that people use. So for one, cell extract, so this would be the contents of your cell. To prepare this, you just wash your cells, lyse them in a buffer like Ripper, you scrape them and centrifuge to remove the debris, and then you can use this or you can store it. With tissue, obviously you need to dissect out the tissue of interest that you're interested in, snap freeze with liquid nitrogen, homogenize, again lyse the cells, centrifuge to remove debris, and then you can use it. Saliva is very simple, you just have to collect samples. Again, centrifuging is just to remove debris, and then you can use it. Serum, obviously collect whole blood, allow it to clot at room temperature, and then centrifuge, but this time with the break off, and again, you're free to use it. Plasma, again, whole blood, but this time you lay it over Ficol, centrifuge with a break, and then use the top layer. Conditioned media, basically, obviously you grow your cells in serum media because they need serum, wash with PBS, and then exchange that media with media that doesn't contain serum, and then incubate and remove the supernatant, and then you can use it. Urine is very similar to saliva, you just need to collect and centrifuge debris out. And then probably the most popular one that most people use is cell culture, supernatant. All you need to do is collect the supernatant, again, centrifuge it because you might have some dead cells in there, and then you can use it. So now I'm going to move on to kind of some things you kind of really need to look at when you're doing your ELISAs, and one of them is your CV value. So your CV is basically the coefficient of variability. So this is very important because it determines on how tight and or how accurate your data is. So the intra-CV is between plate and plate variability. So this means between your biological replicates. So this means your NZ3 of actual biological replicates rather than your sample replicates. You want the variability to be below 10%. And then the inter is between your sample replicates. So this is replicates that you've done within the same ELISA, and you want that to be below 15%. The reason why we recommend always calculating your CV percentage is because it can actually calculate or indicate any inconsistencies within your assay. So this can be through pipetting, it can be through sample handling. You might have allowed the wells to dry out, so you've got your protein or your antibody has been compromised, and it can also demonstrate whether you've got cross-contamination. So it's something that necessarily not a lot of people do, but it is something we recommend doing on a consistent basis because it can show you if there's an error creeping into your ELISAs. So I'll just go into some tips that you can use when you're doing your ELISAs. I've probably said this word too many times today already, but one of them is obviously, the top one is obviously consistency. So I know that the most common practice to use in ELISA plates is what we call uncoated ELISAs or standard ELISAs. This means that you are actually coating the ELISA plate yourself rather than buying what we call pre-coated ELISA plates. We would recommend, if you are seeing there's some issues in consistency, is to try using pre-coated ELISAs because the coating of this is very, very consistent, and probably more consistent than what you generally find in your own. Another one is to make sure your samples are homogeneous. So this means keep them the same. And also try to keep your washes consistent. So another one, which probably sounds really, really simple, but always follow the manual. Now, I know there's some labs that would maybe dilute your primary and your detection antibody. in order to try and get the ELISA to eke out a little bit further. Now, I understand the reason behind that, because obviously you're saving money because you get more ELISAs out of the kit. But please keep in mind that the companies that make these ELISAs, they have worked to try and make the consistency to be as good as possible. So this means they've looked at the ion concentration within the buffer to make sure you've got maximum capture of your antigen and maximum detection of your antigen as well. So it's something that I would recommend. Also try and keep the incubation and temperature times consistent between assays. I'm not saying that we, go into massive climate change in Ireland, like the temperature probably varies between 20 and 10 degrees on average, but just make sure that you keep that incubation temperature the same. So again, you just, like I said before, consistency. Use controls. So what you can do is use a spike, what we call a spike control, to verify the assay. So a spike control basically allows you to make sure the assay is working. So what you would do is you would spike into a sample or into basically just buffer from your samples a known concentration to make sure that the assay is working correctly. Another use for the spike control is to check for a matrix effect. So a matrix effect is basically any effect that your sample buffer might be having on your ELISA. So that can also be important, especially if you're looking at very complex sample preparation. So tissue would be one of them. And always make sure you use the correct negative and positive controls. And validation. Don't go off and buy an ELISA kit and just, stride off and start doing 20 ELISAs, because you might find that you've done that and then you find that your samples are actually falling outside the standard curve, so you can't actually determine the concentration correctly. So always try to validate your assay before starting, and always try to make sure you choose the right ELISA for your protein. So there's obviously many different isoforms of different proteins, splice variants. There's also different conjugations as well. Some might be palmitoylated, some might be carboxylated. So just keep that in mind, that you're choosing the right ELISA for your protein. So that was just kind of ELISAs in a very, very small nutshell. It's probably a bit quick, it's probably a bit brief, but it's giving you kind of like maybe a background on ELISAs. But if you've got any more questions on that, then please just let us know. So I'll just go into a new product that we have from a company called Ray Biotech, and that's for ultra-low detection of ELISAs. So most ELISAs detect within the picogram nanogram range, but they've got this product called an IQ ELISA, which basically combines PCR and ELISA. So it's 10 times more sensitive than ELISA, and it also uses a tenth of the sample volume. So this is critical, say if you've got very, very small sample, or you've got very, very low antigen. So the way it works is very similar to a sandwich ELISA. So you'd have your primary antibody, you'd add your sample to the well, and then you add your secondary antibody, which is actually conjugated to this little DNA strand, which can be identified. You then obviously wash, and then you actually read by qPCR. So the qPCR is amplifying the little DNA conjugate that's onto your secondary antibody. So again, it's very similar to any other sample like HRP or fluorophore. It's just using DNA instead, but it also means because you're doing that, you're making it much more sensitive. So it's something to keep in mind, especially if you've got A, very, very little sample, or B, you need something that's more sensitive. So now I'm going to go into multiplex. So as I said about multiplexing, these can really fall into two camps. So the first camp is bead-based. So for example, this can be using flow cytometry or Luminex, and then the other one is plate or array-based assays. So for example, MSD, Quantis, or Ray Biotech, as I discussed before. So I'll go into bead-based first and give you an example. Funnily enough, I use one from a company that we sell called GeniePlex, which is from ELISA Genie. So the way this works is your beads are basically linked to a capture antibody. Now, the beads come in two different sizes. So this is obviously, the size difference is obviously for visualization purposes only. One size is typically 5 micrometers and the other size is 4 micrometers. and the fluorescence intensity varies in them. So they're actually got, they can be detected by APC, and the intensity of that is depending on which target molecule you're looking at. So for example, this could be TNF alpha, this could be R6, this could be R10, for example. And it depends on the fluorescence intensity, and then obviously use a flow cytometer to measure these. So how it works. So like I said, the beads are detected by APC, and then the detection antibody, which I didn't show in the previous representation, is detected by PE. So when you do your ELISA like you would normally would, and then when you're detecting it on the flow cytometer, you'd first gate, should we say, or separate by using size scatter and APC to identify the target. So for example, this one is bigger and it's got more fluorescence intensity. So like I said, this could be TNF alpha, this could be R10, because it's got less fluorescence intensity. And then from that, you can then, when you have that sample of beads that you've isolated, you can then use PE to identify the intensity. So this is just a relative number of using PE, and then the intensity can tell you how much protein is identified within the well. Don't worry about this too much though, because with all of these companies, they all provide software guides that guide you through this. So don't worry too much about that. So some advantages of using a multiplex array like GeniePlex. A, it uses very little sample. You quantify between 2 to 24 analytes in one assay. It takes very little time to process, just a day. There's off-the-shelf assays, or you can actually customize your panel if they don't have the targets that you're looking for in one assay. It's cheaper than coated ELISAs, and undoubtedly there is a flow cytometer in your university or in your place of work. Sorry. Oh, there we go. Right. Now I'm just going to go into the plate-based array system. So this uses either a slide or a 96 well or a 384 well based system. And the way this works is you capture multiple antibodies onto the bottom of the slide or the plate. So for example, this is what you would do in a normal, on a typical ELISA where you've got a single plex. So this obviously is the same antibody capturing the same antigen, and it's consistent and uniform. While in the multiplex, you've obviously got here different capture antibodies trying to target different antigens. You obviously, again, still require a second detection antibody, and that would be conjugated using either luminescence, molecule fluorescence, or electrochemiluminescence. So as an example, I'm going to give you an example from Ray Biotech, and they're known as the Quantibody arrays. So I'll just go through how this works. So the Quantibody arrays uses an 8-chamber glass slide, but you basically treat this the same as an ELISA. So within each of your wells here, this is your well, you have your array. So in your array, these are basically the different colors are your target capture antibodies fixed to the bottom of the glass slide. So each of these would be different depending on the antigen you're trying to identify. So for example, this would be TNF, this would be R6, R10, blah, blah, blah. And the really cool thing about the Quantibody arrays is the fact that you actually got your replicates within the same well. So this is actually TNF alpha in quadruplicates. This means you've got 4 replicates within the same well. So it means you don't have to use these 3 wells to do your replicates. They're actually contained within the one well. And it's very, very similar to the way you do a sandwich ELISA. As you've got your antigen bound to your capture antibody, then you add your secondary antibody, and then you read it, and then this is what you get back. So you would have different levels of fluorescence intensity depending on how much protein you have here. So for example, TNF alpha is very, very high, but then this target molecule is obviously not present. So some advantages of the Quantibody array. This system uses very, very little sample. You're looking at 20 microliters. The smallest plex they have, so this means the smallest number of targets they have, is 20 analytes, and can actually go up to 100. It takes as little as 4 hours to process. You can either use off-the-shelf assays or you can customize your panel. It's cheaper than coated ELISAs. There's no specific equipment required. There's over 20 different scanners that can be used, and I know that there's at least one scanner in the majority of universities in Ireland. And it's been well referenced, so that it's published in over 1,000 top-rated journals, including Science and Nature. So you might be thinking, okay, they sound really cool, but what's the actual advantages of multiplexing? So I think probably one of the key ones is it saves you on time. If you take example of running 20 different ELISAs on the same target samples, you're probably looking at least a week, depending on how quickly and how organized you are. Maybe not so organized, it'll probably take me a month. But with the multiplexing, you can actually do all of that in just one assay. So just one day. They're cheaper than coated ELISAs. And again, as I said before, coated ELISAs are more consistent than coating them yourselves. So again, you're increasing the consistency, but you're also saving on money. You can also reduce variance between targets. Because you're running the assay at the same time for each target, it means any variance between those can be eliminated. So like I said, if you're running 20 different ELISAs, you're probably having to freeze thaw your sample numerous times in order to get that. So again, you could be increasing the amount of variance between those targets greatly. It can also be used for discovery. So say you have made a knockout or a knock-in and you want to see, oh, so what effect is this mutation or this variability going to have on my cell? So you can actually use this for discovery. So you can see like, oh, you know, what's it done to IL-6, IL-10? It can give you that data. You can also save on sample, like I said before, between 20 microliters on Quantibody and I think it's 50 microliters on the ELISA Genie, so you're saving the sample. So that means you can use that sample for something else. And it can also reduce on errors. Like if you think about the thought of having to do 20 different ELISAs on from the same sample, it actually makes me a little bit sick. But you can also reduce those errors because, I mean, the amount of chance of making a mistake on having to do 20 different ELISAs compared to just doing it once can be reduced greatly. That can be, for example, like mixing up samples or mixing up standards and things like that. I mean, it can have an effect. So I'm just going to recap really briefly because obviously you don't want me to give the whole presentation again. But just some key points. So there is more than one type of ELISA. I always think of sandwich, but there are other types. So you need to choose which one suits your needs the best. So am I looking at hormones? So do I need to be thinking about a competitive ELISA? Always validate your assay first. I cannot stress this enough. It's probably something I didn't do enough in the lab when I was working, and I wish I had, but always try to make sure you validate your assay first. Check your CV values. This can tell you if something is going wrong, and it can be critical because you can go down this rabbit hole and you think you're doing fine, but then you actually find there's so much variability and need to do all of it again. Keep consistent. It's something that it sounds really simple to do. It's actually probably very hard to do in practice, but this will keep your CV values tight, and it means you don't have to keep repeating yourself. Think about multiplexing. If you are looking at multiple targets, it might be something that you want to think about. I mean, even if you're only looking at 3 or 5 plex, I mean, maybe ELISA GeniePlex is something that might be worthwhile thinking about. And in terms of that, which method would suit you best? Some people prefer flow cytometry. I don't understand flow cytometry, so I'd much rather do an array-based multiplex. But Paul, who lives and breathes flow cytometry, would probably prefer to do flow over the array-based system. And don't be afraid to ask. There's obviously not just Paul, Emma, and I that are available, but there's obviously other companies that sell this. So I don't think they'd mind if you asked any questions about ELISAs or about multiplexing. I'd be more than happy to help. So I'd just like to thank everyone for attending. I hope you found it useful. There will be a discount voucher on ELISAs from the companies listed here, available for anyone that's attended. So that's from Ray Biotech. So like I said, they've got the IQ ELISA, the Quantibody arrays. They also do ELISA kits. ELISA Genie, doesn't need to be said that they do ELISAs. I don't think so, but they also do the GeniePlex. So it's the multiplex flow cytometry based. BioLegend, which I think most people probably know quite well, do ELISAs, as well as LegendPlex, which is another flow-based multiplex ELISA, and then Tombow Biosciences as well, which do ELISAs as well. And just let you